back and say, well, there's already 2 other groups doing that, right? So it does behoove you to get it in sooner rather than later. Okay. I hadn't told somebody they had to change, but I did sort of suggest that somebody else already had the topic. Okay. So we were talking about the different parts of the nephron, which thankfully the book has given me a picture, so I don't have to draw it again. So the corpuscle, right, glomerulus inside, capsule surrounding it, that's where filtration occurs, right, by glomerular hydrostatic pressure, which is blood pressure, pushes filtrate out here. Proximal convoluted tubule, we get a lot of reabsorption of water and useful molecules. Distal convoluted tubule, we get a lot of secretion and variability in what is happening there. So we skipped the big long nephron loop because it's the more complicated part. It's not complicated because of what it does. It's complicated because of how it does it. Essentially, the nephron loop allows us to recover a great deal of water and salt from the filtrate before it goes on here to become urine. And because of common location, okay, so most of the nephron is above this line, right? So if we drew a line right here, well, that line is the line between the cortex of the kidney and the medulla being the center and cortex being the outside. So the only parts that are below that line are the nephron loop and the collecting duct. And that's not by accident. So these 2 structures, collecting duct and nephron loop, they share a common region. They're neighbors, okay? Which means that they share a common external environment. So surrounding the nephron loop is an environment. Surrounding the collecting duct is an environment. And it's the same environment. Okay, it's shared. And that'll make more sense when we get there in a minute. Because what the nephron loop does has direct effects on what the collecting duct is able to do. Okay? So the simple thing that the nephron loop does is on the way down, water is reabsorbed, but not salt. Okay, so that's important. Water, but not salt. On the way up, salt is reabsorbed, but not water. Okay, so we have a strange separation in the body where water is doing one thing, salt is doing another, and they're not doing them in the same place. This is part of what makes the nephron loop unique. Okay, so the water reabsorbing part is thin, so we call it, not surprisingly, the thin part, or the thin half. And then the thick loop is where sodium is reabsorbed. Okay, and like I said, I'll show you some better pictures here in a minute about how that happens. But essentially, you know, this is how the salt part, so we're up here in the thick limb, salt is moved through active transport. Okay, so these carrier proteins are pumping this sodium and chloride from the tubular fluid into the peritubular fluid. So it's and then into the blood eventually. So what happens is, you know, remember, water follows salt. I'm going to write that up here again because today's lesson is really all about this. Okay, water follows salt. And incidentally, this is usually because of osmosis. Everybody's favorite topic, right? Osmosis is notorious for being non-intuitive, difficult to understand. But essentially, osmosis in a nutshell is movement toward solute. So it's movement of water toward solute. Now, that solute could be anything, but in this case, we're talking about salt, sodium chloride. Okay? So, in the descending limb, water moves. Ascending limb, salt moves. This is just how the salt moves. Okay, so one of the reasons that this structure is so important is because it develops, it creates in the kidney an osmotic gradient. Okay, so now we take 2 concepts that are not always intuitive, osmosis and gradient. Well, a gradient is just a difference across a region, okay? the strength of osmosis, or the strength, what makes osmosis happen, is what we call osmolarity. Osmolarity is how much stuff is dissolved in fluid in a place. Okay, so think about sugar and water. If you take pure water and you add a little bit of sugar, you have a low osmolarity, right? Because there isn't much solute in that sugar water you made. So there isn't a pull towards that solute. There isn't an osmotic pull. Now, if you took a whole cup of sugar and put it in your water, now you've made a thing that has a lot of osmotic pull. So it has a lot of osmolarity, we call that. Okay? So, as the salt is pumped out of this tubular fluid, well, salt is solute, right? So the solute concentration out here gets higher and higher. You're pumping salt from one place to another, so and you do it more down here than you do up here, so you get this osmolar gradient. These numbers are osmolarity, how much stuff is dissolved in the fluid that is surrounding this nephron loop. Okay, so this is the nephron loop, and all this space around it is peritubular fluid. Okay, and this is one place in the body where the tissues here have very different characteristics than tissues everywhere else do. Okay, normally in the body, the osmolarity is 300 or above. This is one of the few places where we get very high osmolarities in the tissues of the body, and that's all part of how this system works. Okay, so let's look at what's happening in the fluid in the nephron. It comes in at about 300, okay? That is coming from the proximal convoluted tubule. So a lot of water has been reabsorbed. A lot of sodium has already been reabsorbed too. Glucose, amino acids, fats, things like that. Okay. Well, as we go down, water leaves. See, water leaves. Well, if you take a solution that has an osmolarity of 300, and you take water away from it, its osmolarity is going to get higher, right? Sugar water again. If you put your sugar water on the stove and you boil it, it gets more and more sugary, right? Its osmolarity gets higher and higher because water is leaving. Same thing happens here in the descending limb. Water is leaving, so we see the osmolarity get higher and higher and higher until we get to about 1200 here. And what pulls that water out is the solute that is present around this tube. Remember, water, we can't move water. Water has to move towards something in order to move. In this case, the water in the nephron loop is moving out towards the higher osmolarity in the peritubular fluid. Okay? Now, well, where did this solute come from? It came from the salt that's about to leave next. Okay, so we see the solute gets, or the filtrate gets more and more osmolar, right? The number gets higher. Turns the corner, starts to go up. Now we see the numbers getting lower. Why? Well, now the salt is being pumped out. So in your sugar water, if you started taking the sugar out but leaving the water behind, tricky to do in the kitchen, but the kidney can do it because it's molecular machinery, right? So the salt is pumped out, leaving the water behind, and we see the numbers get lower again. Because, you know, why? The osmolarity is being pumped out here. That same salt is then going to be what draws the water out from the descending limb. Do you see that? So the two sides are linked. The ascending branch pumps the salt out. That salt draws the water out of the descending limb. Now, all told, if we look at this whole picture, we see that water has been recovered and salt has been recovered. Two things our body always needs, right? So it is a way to recover water and salt from the urine. Now, in addition, it's a way to create this gradient, which the collecting duct is going to pass through this gradient too. And I'll show you why that's important here in a minute. But we call this countercurrent multiplication, is the technical term. And what that is talking about is that the salt pumped out here draws water out from what's coming in. So you get kind of an amplification effect because the two, you know, the fluid is entering in one way, leaving in another, but it's being massively altered in between by this water moving. Okay, so water and then salt And the real key here is this osmolar gradient. So here at the top, this is the cortex, right, of the kidney. This is deep medulla. And as we go down, we get more and more osmolarity in the peritubular fluid. All right, so questions about that? Okay. Yes? Right. Because we're going to now take this and we're going to use it to do something else. So all told, urine or filtrate goes in at 300, it comes out at 100. That's pretty dilute for urine. Okay, if that's what entered the bladder, we would have a very high urine output of very dilute urine. Okay, so it's got a lot of water in it, but it doesn't have very much waste products in it for the amount of water it has. So those of you that follow good water hygiene and you drink a lot of water every day, your urine stays at about this 100. So you're not doing a lot of extra water recovery. And you can do that because, let's face it, water is very plentiful in our environment. You know, it comes out of the taps, so you can drink plenty of it. However, that isn't how we evolved. You know, we evolved to be water conservers, you know, because our ancient ancestors lived in a variety of different environments, some of which did not have lots of fresh, clean water to drink. So the story doesn't end there. Because now we're going to use this gradient. Okay, to orient you, this is proximal convoluted tubule, right? Here's the descending limb. See how water is leaving? Here's the ascending limb. They don't show it, but salt is being pumped out here. Here's that gradient represented by color. So the more red, the more osmolar it is. This is the distal convoluted tubule. And then this whole thing is the collecting duct. And this is one of my favorite pictures in the book because of the little circles. It's a visual demonstration of concentration of different things. So it isn't just for coloration. Each of the circles is representing something. And how many circles there are crammed into an area is sort of the lesson here. Okay, so the purple circles are sodium. The green are chloride. You can ignore those because chloride is pretty much inert. And then the urea ones are in brown. And remember, urea is the waste product. That's what we want to get rid of. Okay? And in addition to urea, you could also add creatinine, ammonia, hydrogen ions as waste products too. Okay, so coming out of the proximal convoluted tubule, we have an osmolarity of 300. What does that look like? Lots of water with a few circles in it. Okay, so it's pretty dilute still. Now, as we go down here, the water leaves, so the solutes get closer together, which you can see that. You know, they're more packed together. Now, as we go up, the purples are pumped out. Okay, so they're pumped out here. So by the time we get up here, there's many less purples. But the oranges, the urea, stay in. They're never pumped out. Okay, the urea that comes in here is still in here, is still in here. So even though the water is being removed and the salt is being removed, the urea is not being removed. It's staying behind. Okay, so now we come back up here. We have very dilute. Actually, this should look more dilute than this, but it doesn't in this case. And we make this bend and we go back down. Now, why is that important? Look at the difference between how packed they are here versus how packed they are down here, right? This is much more concentrated urine than this is, certainly more concentrated than this is. Now, how does that happen? The collecting ducts have channels that are either open or closed for water. And the controller of those channels is, it's got to be out here somewhere, is ADH, is antidiuretic hormone. Okay, so these dotted lines are channels controlled by ADH, antidiuretic hormone. So when those channels are open, as the fluid passes through the collecting duct, it passes through this osmolar gradient again. The same one that was made in this nephron loop through that countercurrent system that I just talked about, where the water comes out and the salt comes out. Well, as the urine passes through here, if water can exit, the concentration in here, the osmolarity, is going to be the same as it is out here, because things are going to move by osmosis. Water's going to move towards where the greatest solute is until they're in balance. So when these channels are open, which is what this diagram shows you, you see the 300s match, the 900s match, the 1200s match. So water's going to leave until the concentration in here is the same as the concentration out here, which is very, very concentrated. So this is many of us first thing in the morning. We haven't had anything to drink for 6 to 8 hours, so the body is a little bit fluid deprived, and the urine in the morning is usually very concentrated. That is the effect of ADH. When ADH is present, these channels are open, allowing water to exit the urine and the urine to become more and more concentrated, have more waste products in it for the amount of water that's present. Okay, so like I say here, this is a key water conservation strategy for the body. And note that urea, how much urea is removed, doesn't change. It's just the amount of water that goes with it that changes. Okay, so across the day, minute per minute, the kidney gets rid of about the same urea all the time, even though our urine output changes according to how well hydrated we are. So if you drink a lot of water, your urine's going to have a lot of water in it with urea. If you drink a little water, you're going to have a little water with the same amount of urea in it. Okay, because that's how this system works. Okay, so water control is essentially what this is. Okay, so this kind of water recovery, where these channels can either be open or closed, we call that variable water recovery or regulated water recovery. Because there's essentially two ways we reabsorb water. One is what we call obligatory. In other words, it happens all the time no matter what the body's water status is. And the other is facultative, which those of you that have had microbiology will have heard that word before. That means it can be like that or it cannot be like that. It's controllable or changeable. So in the first half of the nephron, it's all obligatory. So we get a bunch of water recovery in the proximal convoluted tubule. We get even more in the thin descending limb. And then when we get over here, there is no water reabsorbed here, only salt. But in the distal convoluted tubule and the collecting ducts, we have variable water recovery. So the key player to all of these dotted lines is ADH. So in our first chapter together this semester, we talked about antidiuretic hormone. And I said it was a desert hormone, right? Desert hormone, ADH. So when you're, you get that? A desert hormone, ADH. So what does that mean? It means when you're dry, ADH is released, and it causes the body to hold on to water. Now you know how. The body holds on to water by opening these channels in the distal convoluted tubule and the collecting duct to allow water to flow down this, into this osmotic gradient and out of the urine. Eventually, the water in this peritubular space ends up back in the bloodstream and not in the urine. That's the point, right? Because in the desert, you don't want to have a high urine output because that's water you're going to have to put back. So it works at these water channels. The water channels are called aquaporins, which it usually says somewhere in here. But maybe it doesn't. So you don't have to, if it's not on the slide, it's not something you have to know. So variable water recovery. All right. Okay. So ADH is, because it's a hormone, it means that the levels can vary, which means that this system can be turned off or on or up or down. It's not like a light switch. It's more like a dimmer switch, right? You can make it very high, very low, or somewhere in the middle. So this diagram, it shows you the difference in the physiology between when ADH is present and when no ADH is present. So people think that evolutionarily, this came first. Because, at least many theories say complex multicellular life originated in the water, right? It originated in the ocean. Where in the water recovery or water conservation is superfluous. You don't have to worry about keeping track of water when you live in water, right? So the kidney evolved first like this, to produce a large volume of very dilute urine. So this is what we see in fish and invertebrates, any creature that has a proper kidney, right? So we have, urine is produced, but what comes out, large volume of dilute urine, because there's no further change after the distal convoluted tubule, right? So we get the proximal, the nephron loop, distal convoluted tubule, and then this urine looks just like this urine that leaves the animal, okay? Now, let's add ADH to that. So over time, there evolved on the collecting ducts and the distal convoluted tubule these water channels that open when ADH is present. So we open those water channels, and look, a whole bunch of water leaves because this sodium has been pumped out, making this gradient, pulling this water out of the urine, so we get a small volume of very concentrated urine, and everywhere in between. Okay, so people that drink a gallon a day or more, right? They have very little ADH circulating, so their urine looks like this. People who don't drink water enough, they have very little ADH circulating, which looks like this. Okay. Now, ADH is involved in a couple of different disease states too, which you all will learn about in a future class. But there are problems in both directions. So a disorder called diabetes insipidus. So there's really two kinds of diabetes. Diabetes mellitus is the one we all hear about that has to do with insulin and blood glucose, right? Diabetes insipidus results from the absence of ADH secretion, either from brain trauma. This happens, so in a bad car accident, closed head trauma, you can get diabetes insipidus. It can also be a genetic problem. It can also be a post-viral problem. But if you don't have any ADH, what are you going to do? Well, you're going to have a large amount of dilute urine, regardless of your hydration status, right? So if you don't make ADH, this is what you're going to look like all the time. So what you see in diabetes insipidus is patients have a very high urine volume, even though they don't drink very much, okay, which means they often run into dehydration. However, in the conscious animal, if you have a high urine output, you just drink a lot too. So these are people that drink a lot and pee a lot. But the difference is, what happens if they stop drinking? In the diabetes insipidus patient, their urine output stays high. So it's not associated with their water intake. So that's one. You also have this. You have a syndrome called SIADH, Syndrome of Inappropriate ADH Secretion. Again, you can get that from closed head trauma. This is where you make ADH when you don't need to make ADH. So you have patients that end up in volume overload. They have too much circulating blood volume because they're holding on to every little drop of water from the kidney. So oftentimes those patients need dialysis or at least careful fluid management. So you'll learn about those things later on. But ADH is one of the primary regulators of water in the body. And as I say down here, we don't live in the ocean. The ocean has a pretty consistent salinity. In other words, for the water that's present, there is pretty much always the same amount of salt. When animals left the ocean, they faced this challenge. On the ground, water and salt are not always in the same place, right? Salt is usually found in minerals or in plants, and water is found on the surface. So we need a system that manages water separately from salt, because on a given day, your water intake and your salt intake are going to vary, and they're not going to vary together, right? We don't drink water that is pre-salted. If we didn't have ADH, we would have to. Our drinking water would always have to have the right concentration of salt in it. So because ADH affects water without affecting salt, it allows us to have salt intake that changes and water intake that changes and not have them be related. So it may not seem like a big deal, but if we didn't have ADH, our diet would have to be totally different, and we'd have to think about it a lot more than we do. So it is convenient that we have this system that can either dump excess water or, in this case, dump excess water or hold on to excess water according to our diet. So enough about all that. Okay, another one. This chapter is full of really great slides or pictures from your book. That's why I keep saying, oh, I like this one too. Nephrology, which is what you're beginning to study, or the study of what the kidney does, is notoriously challenging information because it includes some biochemistry, it includes some regular chemistry, a little bit of physics, all mixed up into this complicated thing. Your book does a pretty good job with taking it apart and presenting it at a real basic level. But this is another one of these slides where I say this is a great study page because it puts the last 2 days of me talking really onto one big sheet of paper. Okay, so I'll show you what I mean. It all started at the glomerulus, right? Blood went in. This is the afferent arteriole, efferent arteriole. There's pressure in here that creates filtration, right? So fluid is pushed out. That fluid moves into the proximal convoluted tubule where nutrients are reabsorbed, water is reabsorbed, and electrolytes are reabsorbed, right? So that's all right there. Where do those things go when they come out of this fluid? Well, they're going to go into the peritubular capillaries, which are these guys, all right? Then if we continue to follow what's becoming urine, it goes down the descending limb, a bunch of water comes out. Where does the water go? Into the peritubular capillaries again. That comes down here. It's gotten very concentrated. You see that with the circles. Goes back up. Now the salt gets pumped out. As the salt gets pumped out, we create this gradient, right? The osmolar gradient that I was talking about. Back up to the cortex. Distal convoluted tubule. Here's an aldosterone pump, pumping sodium out, potassium in, like we talked about last time. And then as we go down through the collecting ducts, here are ADH-regulated aquaporins, or water channels that are either open or closed, depending on fluid status. Another point to look at is if you follow the blood here, it goes down, it follows the nephron loop down, and then back up. And as it does so, okay, so here's our osmolarity numbers. Okay, if you look, the blood's osmolarity is matching that. Now, typically, the osmolarity of blood is about 300. So this blood in this part of the body is 4 times as concentrated as anywhere else. If you took this blood and you just dumped it into a vein, it would cause cells around that site to, they would shrivel up because all their water would be pulled towards this very concentrated blood. That's bad, right? So in order to keep that from happening, so here we have blood that's at 1200 because the surrounding tissue is at 1200. It goes back up the nephron loop before it goes back out to the rest of the body. So this very concentrated blood draws out this water that's coming out from the descending limb, and that dilutes it. So see, here's 1200, it gets some water. Here at 600, it'll get some more water, and eventually it'll leave at about 450. Well, that's way better than 1200. So this system is the vasa recta system that I introduced in our anatomy introduction last week. Okay, so connect this with that vasa recta slide that had the juxtamedullary nephron, big long loop, regular nephron, cortical loop with a little, or cortical nephron with a little loop. Here's the connection to that. Okay, and it talks about that system right here. Okay, a lot of good stuff on that slide. Okay, so now a little bit of sidetrack. You know, I never test you on the disorders, but they're good to talk about anyway. Yes?
I have a kind of question that's related, I guess. I took my dog to the vet this morning, and he's a puppy, so he peed in there, and they like tested it and saw that it was like all clear and that it was just above the concentration level of water, so like it's basically just water. And they told us to watch it and like to see.
Does he drink a lot?
He does, yeah, but she said to watch it because she said that his pee might not be getting concentrated, but I didn't know.
They're talking about diabetes insipidus.
I thought that's what that was when you said that.
Yeah, DI, diabetes insipidus is what they're worried about. You'd have to look at there's 2 variables to look at. One is urine output. One is water intake. Because you have to look at both. If you drink a lot of water, you're going to have a lot of dilute urine. And not only just like people will, we call it psychogenic. People will drink water all day because they like the way it feels in their mouth and stuff. Dogs will do the same thing.
Yeah, he drinks a lot of water, but he pees a lot.
Yeah. The one thing to test, I mean, listen to your veterinarian, but the way to test it would be to take the water away and see if the urine output goes down. Because in diabetes insipidus, it won't. Now, the danger is now you have high urine output and very little water intake, so dehydration becomes an issue. But that's one of the ways you test it.
So that's what
in
she
real
meant,
life.
like it might not be getting concentrated?
Yeah. In diabetes insipidus, this doesn't happen right here. So the urine doesn't get concentrated, so we get this. We get a lot of large volume of dilute urine. Good, excellent connection, just what we're talking about. I hope Puppy's fine, though.
Yeah, he's had problems ever since we got him, so it's been.
What kind of dog is it?
It's a black lab.
Oh.
He just keeps having stomach and urine problems.
Oh, poor thing.
Yeah.
Well, hopefully he doesn't have something serious.
I know.
The pet dogs they've been bred selectively for so long that sometimes they do have a higher incidence of genetic disorders.
Mm-hmm.
But they also are quirkier than their wild counterparts, so he may just like to drink water.
He does love to drink water, and I'm not,
Well, and labs are water dogs anyway, so just being around water may be just special for the black lab, I don't know. All right. So a little bit about renal failure and dialysis. So essentially, renal failure is, not surprisingly, when the kidneys fail to do their job. Specifically, that's getting rid of waste products. Now, kidneys can fail at the other things they're supposed to do too, like managing water, but typically we don't call that renal failure. We call it whatever the underlying cause is, like we were just talking about. Diabetes insipidus, the kidney isn't doing its job, but it's not renal failure. Renal failure is the inability to get rid of the waste products. And it comes down into two varieties. Chronic renal failure comes on slowly over time, usually from some chronic illness. So a while back we talked about how diabetes mellitus is the leading cause of chronic renal failure, preventable renal failure in the world. And that's an example of a disease causing changes to the kidney which result in chronic renal failure. Now, there's other versions of this. There are some genetic forms of chronic renal failure, like the polycystic kidney disease. There's some acquired ones. Streptococcal, post-streptococcal glomerulonephritis can cause this. So this is long and slow, basically. This happens over years. Acute can happen over days, even hours. And acute renal failure usually results from some ischemia or a toxin. So let's say somebody isn't paying attention and a patient is given a dose of gentamicin, which is an antibiotic, that's 100 times greater than what it should be. They will, by the end of the day, be in acute renal failure because that particular antibiotic attacks kidney cells and causes them to shut down. Another version of this is somebody is walking along, they have a heart attack and an arrhythmia, and they drop dead, no circulation, right? And they're brought back some minutes later, heart starts beating again, but the kidney will have been affected by that absence of blood flow, and that can result in acute renal failure. So, like on TV, it's exciting always when they get the patient back, with the paddles and the epinephrine, everybody's hollering, it's a big happy moment or whatever. Well, oftentimes days later, they'll find that the patient has acute renal failure and may die anyway. Or may need dialysis for the rest of their lives until they can get another kidney. So it isn't always over. After the brain is the most sensitive organ to oxygen deprivation. The kidney is the next most sensitive organ. So even if the noodle survives, sometimes the kidney, one or both, don't. So that's the difference between chronic and acute. Dialysis is our, not entirely unclever. I mean, it's a clever system, but it's no substitute for the real thing. Dialysis is essentially mechanical kidney, doing with machines what the kidney does. And unlike a cardiac bypass machine, where it's really just a pump, right, because that's what the heart does is move things around, well, what the kidney does is far more complicated than that, as you've seen. You're talking about molecules moving, water moving. We have a lot of complex things happening in what the kidney does. So in the dialysis system, we take blood out of the patient here, and we run it through a pump, and we put it through a special tube that is surrounded by a special fluid. That tube is what we call a semi-permeable membrane. Some of you will have worked with dialysis tubing in a lab in some point in your life. You know, where maybe you put it in a bag and the bag got bigger or smaller. Sound familiar to anybody? No, it's a classic lab, but I guess people don't do it anymore. But anyway, this tube allows water to pass, it allows small molecules to pass, but it doesn't allow large molecules to pass. Okay, so as the blood passes through this fluid surrounding it in this special tube, the small molecules exchange, they go across, while the large molecules like blood and proteins stay in here. So that the blood that goes in here is different in consistency than the blood that comes out here and then goes back into the patient. So using this process, we can get urea out. We can manage glucose levels a little bit. We can manage sodium and potassium levels pretty well. The problem is this is a slow process that takes over people's lives. In advanced renal failure, it is not uncommon to need dialysis 3 to 4 times a week. Usually a dialysis session is 2 to 4 hours. Any dialysis techs in here? No? Okay, sometimes there is. So, I mean, think about that. You're going to be at that dialysis clinic more, you know, you're going to feel like a job, only you're paying them, right, to be there. So it can be really, really life-changing for people. Now, it's life-saving, and it's a way to get a person by until they can get a replacement kidney, if that's possible, and if the disease they had that killed off their first kidney wouldn't kill off the transplant. We don't do that. You know, so for example, if you have an autoimmune disorder that destroyed your kidney, you can't get another kidney because you'll destroy that one too. So you're left with dialysis as your only option. Now, another option that we use, particularly in children, is something called peritoneal dialysis. And this is where we don't use a machine to move blood around. Instead, we use the patient's peritoneum, their abdominal cavity, as a dialysis chamber. So essentially, in peritoneal dialysis, we take this dialysis fluid and we put it into their abdominal cavity. So remember, there's the abdominal wall, and then there's the digestive tract, and there's that space in between, right? We fill that space with dialysis fluid. And, you know, because there's circulation through the digestive tract, blood is traveling next to this fluid that we've put in all the time. Essentially, we've recreated this system right here without the pump. The heart's the pump. So we put that fluid in, then we have to pull it out, and we have to put it in again. So we have to keep exchanging it in order to make the molecules move. So in babies in particular, in kids less than, 10, they do it at night. So they'll put dialysis fluid in and out 3 or 4 times while the kid's asleep. And then when they wake up, it's like they've had a dialysis session. So that's peritoneal dialysis. It's a little bit safer. The problem is you're putting fluid in and out of a body cavity, so the risk for infection is very, very high. But it is something that it can be done at home a lot of times, and some of you will take care of sick kids with that system happening. So, you know, so it takes a long time. What else goes wrong with this? Well, you can imagine that to get enough blood out of the body, 2 to 4 hours a day, 3 days a week, that's going to become a problem at some point, right? Because blood has a tendency to clot. And as many of you will learn, the thing that clots first is anything man-made, all right? Our vessels have systems for preventing blood clots from forming. IV tubing does not. Plastic does not. So anytime you have a line in a patient, in other words, a catheter in a vessel, that's going to be what clots first, not their vessel. It's going to be the plastic that clots. So in dialysis, what they do is they create what's called a shunt. So they take the radial artery and they attach it to one of the large veins that accompanies the ulnar artery on the other side, and they essentially make this easy path for blood flow. And then when it's time for dialysis, they stick a needle, not in the vein, but in this plastic tube that attaches the artery and vein. So we call this a shunt. And accessing the shunt, you put they're wide bore needles. They're great big huge things. You stick them in there, one on each side, and then you have an in and an out. So if you take care of a patient who's on dialysis, ask them where their shunt is. And they'll tell you, because they'll know, because people stick needles in it all the time. And take your stethoscope and listen to it, because it's a unique sound. Because you hear blood flowing directly from artery into vein through a large tube, which doesn't happen naturally, right? It always goes to capillaries and back. So you hear this whooshing sound. It sounds like a dishwasher in the shunt. Now the problem is these don't last. These only last for about 3 months, and then they clot off. And you only have 2 arms, right? So you get to do that twice, and then they have to get creative. So you get 1 in each arm, and then there's some places you can do this in the lower extremity. But the bottom line is, once you're out of spots, that's when dialysis starts to become an ever-increasing problem, because you run out of places to put shunts. So, because eventually when this clots, these arteries and veins scar, and you can't put another one in. Now, sometimes you can move it up. You can keep moving it up closer, but sometimes you run out. Because you've had some experience with this, haven't you? No. I thought you had somebody on dialysis. I think you have somebody.
No, it was
a friend.
Yeah.
Yeah. So shunts, dialysis, and fancy biochemistry through semi-permeable membranes. All right. Let's do some questions, and we'll call it a day. The primary function of the proximal convoluted tubule is what? And I did, again, I forget my phone. Maybe if you do ding me on my course evaluations, I'll remember. I apologize. So what do you think? Yes, D. When you think primary convoluted tubule, think reabsorption of important stuff. So that would be ions, organic molecules, vitamins, and water. Good. Filtration only occurs where? Good, the glomerulus at the capsule, right? And secretion of drugs, acids, and ammonia, where does that fit? Yeah, distal convoluted tubule. All right. ADH, antidiuretic hormone, does what? Just think through its name. Remember, a diuretic increases urine output. Good. C, increases the permeability of the collecting ducts to water, allowing water to flow down that osmolar gradient into that gradient space and out of the urine. So we get a small volume of very concentrated urine. Now, where does ADH come from? Anybody remember? Yeah, posterior pituitary, right? It's a neurohypophysis. And it's released in response to osmolarity. So as your water, let's say, okay, you start at baseline and you very quickly drink a liter of water, right? Water is very rapidly absorbed in the digestive tract. So if you take a solute, your salt water basically, right? And if I add a liter of water to salt water, it gets less salty, correct? So its osmolarity has gone up or down. Down. Because you had more water and less stuff. So less osmolar pull. So as your osmolarity starts to fall because you drank a liter of water real fast, your ADH level goes I gotta think it through. Your ADH level goes down, which causes less permeability of the collecting ducts to water and a higher urine output then. All right. Okay, and we'll call it a day right there. We'll finish with the